Okay, let's continue. Okay, before I go to data analysis for single-cell ATAC-seq, I want to emphasize the importance of experimental design to avoid batch and confounding effects. This is the same for any experiments. In previous homework, you worked on a batch effect correction problem for bulk RNA-seq data. And Martin, your TA, also includes this slide in the single-cell RNA-seq lab. Before you start any experiment, always involve a statistician. This will help you to avoid such problems. As the statistician Ronald Fisher said, to consult the statistician after an experiment is finished is often merely to ask him to conduct a post-mortem examination. He can perhaps say what the experiment died off. Right. So really you should have a balanced experimental design and also can mix different groups of comparisons that you want to make, mix them together for different batches. So after, say, for example, after you obtain the data and do a PCA, you know this is good because the same kind of samples that cluster together, even they're from different batches. For single cell studies, now one can do multiplex and barcode before different samples and then run them together within a single 10x controller, and that can help to mitigate this batch effect. Before I go to single cell ATAC-seq analysis, I want to just summarize the single cell RNA-seq analysis. This is a typical workflow. In the single cell RNA sequencing lectures, you also see this slide. So we usually get the sequencing data off the sequencer and then align them to the transcriptome and do quality control, normalize the data somehow, and then do dimension reduction, whether it's like PCA, t-SNE, UMAP, and then cluster cells and do cluster annotation. And there's some downstream analysis that you can do, trajectory inference, differential expression, and compositional analysis, etc., etc. The single cell ATAC-seq analysis follows a similar workflow with some special treatment for the count metrics, since the data are much sparser than single cell RNA-seq. So as you can see, there are many steps in the workflow. And to streamline the analysis, our lab Shelley's group developed a tool called Maestro. And to address these challenges of processing single cell data, we developed this tool Maestro. Maestro is an integrative analysis workflow based on SnakeMake and for both single cell RNA-seq and single cell ATAC-seq data analysis. So you're going to use this tool for your homework 5. I believe some of the students may have already tried it. If you are not familiar with SnakeMake, I would suggest to you to look it up on the internet. It was developed by Johanna also, who was in Cherry's group as well. And this SnakeMake is gaining popularity in the bioinformatics field for writing workflows, and it's Python based. So if you know Python, you can learn it easily. Okay. So it's a SnakeMake pipeline with steps ranging from pre-processing from FASTQ files and do some quality controls, QC, and downstream analysis, including clustering, differential gene expression or differential peak enrichment, cell type and annotation, and the regulator identification. So it also supports integrated analysis for both single cell RNA-seq and single cell ATAC-seq data if you have samples run through both assays, or you can also handle the multi-omic data as well. So I will explain the features in the following slides in a little bit more detail. So, okay, the first step when you get the data, it's always align the data into the reference, whether it's a transcriptome or genome, right? For Cell Ranger is a 10x genomics solution, so it's from the company. It can do the single cell RNA-seq analysis, also single cell ATAC-seq data analysis. The problem of that is it's kind of slow. It may take maybe over like 20 hours to just process a medium-sized data. And for RNA-seq, actually we use this tool called STAT-Solo. It's much faster, and it's 10 times faster than Cell Ranger. So we use this tool in Maestro. And STAT-Solo was developed by the same author of STAR. I believe you used STAR for bulk RNA-seq analysis in your previous homework. For ATAC-seq, there are different aligners that you can use. A BWA, which is popular, was developed by Han Li, and Minimap2 is also developed by Han Li in our department. It's 15 times faster than Cell Ranger. And I believe you also, in the previous lectures, you also used this called pseudo alignment tool to do quantification for RNA-seq data. The question here is that can you use Salmon or Callisto, those pseudo alignment tools for single cell ATAC-seq data? Can you use that? So think about the difference between ATAC-seq and the RNA-seq. Right. For RNA-seq, you know the features that you're looking at, right? So for example, in human, we have 20,000 genes. You can count how many reads aligned into each gene. But for single cell ATAC-seq, you don't know what to start with. You want to call-peek and then find those open chromatin regions, then count number of reads in each peak. But actually, you can also do that if you have a public available data set which combines all the DNase1 or bulk ATAC-seq open chromatin regions. You can consolidate them into a big, more like a reference. So you can count number of reads in those potentially open chromatin regions. And using that, you are still able to do like clustering, finding new clusters. But again, any reads that not aligned into those public available potential open chromatin regions will be missed. So you can still use it, but then you lose some information. And of course, sequencing has errors. Right? You will need always to do barcode correction. And for 10x, they have their they call it whitelist. So they know the exact sequence they put into the gel beads. And after sequencing, you can compare your sequencing results, the cell barcode, out from the sequencer to the whitelist, and then you can correct based on the Hamming distances. Okay. For bulk level sample QC, that we always call peaks first using Max2. And this is by collapsing all the cells together as a pseudo bulk, and then you can call peaks using Max2, which is a popular algorithm for ChIP-seq data. And it was also developed in Shirley's group. And for bulk RNA-seq, we use RCQC and I realized they actually just recently published RCQC2 for faster quality control for RNA-seq data. And there are some other standard ChIP-seq QC metrics for ATAC-seq, for example, the number of high quality peaks, fraction of reads in peaks or promoters at the bulk level. And an example here is that so you can do a bulk cell ATAC-seq, and also you can do single cell ATAC-seq side by side, and you will see you can kind of collapse. So here, each row here is a single cell, and each black bar here is one read. So if you kind of aggregate those reads together, it should give you a similar profile, just like doing a bulk cell ATAC-seq. So for technology development, quality control, one can compare the consistency between replicate runs, right? For example, here, for this particular cell line GM12878, you can run 2 times, one batch with 200, around 300 cells, the other batch with 4,500 cells. And then here, each dot here is the peak location. So then so you can plot a correlation to see log 2 transform the count per million to see how well those 2 batches are correlated. So a successful experiment will have a high correlation. This is another Baryon experiment in which we have mixed mouse and human cells, just like in single-cell RNA-seq, as introduced by the last lectures. So A20 is a mouse cell line, and GM12878 is a human cell line. So so ideally you expect, okay, those cells are only from the mouse cells, and those cells are only from the human cell line. And for those dots, those are doublets, which contains reads mapped to both human and mice. And as expected, if you overload the 10x machine, you start to get more doublets. So the number of cells per droplets actually follow a Poisson distribution. To minimize the number of cell doublets, the single cell suspension is loaded into the microfluidic device at a very low concentration, so making it very unlikely to have 2 cells enter into the same droplet. But one thing is that most of the droplets are fully functional. They contain the barcoded beads and also the reagents, but they do not receive a cell, and therefore they are wasted. So there are other methods, for example, cell hashing by labeling the cells from different samples with a barcode first, and you can pull cells together and overload the 10x machine. I think the recommended maximum number that you can load into a single 10x lane is like 10,000 cells. But with this cell hashing or pre-barcoding strategy, you can overload the cells. You can overload the cells into the machine. And however, this is not necessarily used in the common experiments. This is really just for the technology development. Okay. Now we go to the real QC for each cell. And we know most of the promoters are open, and we should have enrichment of ATAC-seq signal in the promoters. Right, so you can either have percentage of reads mapped in the promoters or percentage of reads in the peaks. The higher the better for a good experiment. And if you, for example, if a higher percentage of reads mapped to mitochondria, just like in single-cell RNA-seq, that's not good. But the exactly cutoff, for example, the mitochondrial rate need to be determined by each experiment. So each experiment may have different level of mitochondrial contamination. So for here, this is the Q for this quality control here. Like for the x-axis, this is the total number of unique fragments in each cell. So here, each dot here is 1 cell, and the y-axis is the TSS enrichment score. So you can, this TSS enrichment score was defined by ENCODE for bulk RNA-seq, for bulk ATAC-seq, and here we just borrow it for single-cell ATAC-seq. And if you really want to know the details how to calculate this TSS score, so you can go to this link here. I wrote a blog post about how to calculate this. And so in this example, you see, okay, people usually use like 1,000 fragments, unique fragments as a cutoff. And here they use, I believe it's TSS score of 8 as a cutoff. So those are the high quality cells. This is for both human and mouse, so similar. And in Maestro, we actually use this more simpler metric. It's called fraction of reads in peaks. So, and essentially we get the similar pattern. So those blue cells are high quality cells with high number of uniquely fragments in each cell here. and also high fraction of reads in the peaks. Although it seems a lot, like those cells are a lot, but those cells are low quality and they contain very little number of reads. So you still get a good number of reads actually mapped to those good cells. And if you do a pseudo bulk again, using only those cells, it will have a better like a fraction of reads in peaks after this cell quality control. Okay. So what else could go wrong? So I mentioned that there could be a doublet problem. So more than 2 cells, they're in the same droplet, although they are 2 distinct cells, but they receive the same cell barcode. And after sequencing it, we can't differentiate whether they're from same cell or 2 cells. And the other problem is that different barcodes actually can be from a single droplet. This is a paper published in 2019 by Caleb, who was our previous TA in last year. And here, within the single droplet, it could be you have you can have like 2 beads which contain different cell barcode. Or you can have a single bead, but within that single bead, you have 2 different barcodes. So roughly like 5% of the beads contains multiple barcodes, and 4% of the droplet contains multiple beads. And this is only happens for single-cell ATAC-seq, but not for single-cell RNA-seq. This is the 10x solution. And then this brought attention for them, and they later fixed this problem. So the idea is that you don't trust the data other people give you, or also you don't trust the specification that whatever the commercial solution tell you. But you really need to do a lot of sanity check or investigation, some detective work. Okay, this is another Zoom 4. So the question here is, which one is better for quality control for single-cell ATAC-seq? One is fraction of reads in peaks. Second is fraction of reads in promoters. Any answer? Which one is better? Or maybe there's no better or worse. Let me see the pool. Okay. Now, personally, I think the fraction of reads in the promoters will be better than the fraction of reads in peaks, because it really depends. For the first metric, it really depends how many peaks you have, right? Because the more peaks you have, the higher fraction of reads will be in the peaks. And the number of peaks in a pseudo bulk, that can vary a lot by tuning the parameters, right? If you lose the stringency a little bit, you can get a lot more peaks, and then you get a higher fraction of reads in peaks. But fraction of reads in promoters are relatively constant, right? They're there. So I tend to use fraction of reads in promoters. So I vote for that.